So I'll just finish up really quickly with the raster that we were looking at. Just to remind you, this is mean annual temperature, and now you know how to check what information is available, the metadata associated with the raster, its spatial reference system, its resolution, all of that. So whenever you bring in any raster or any other GIS layer for that matter, please always check the metadata, okay? So you know who generated the data, see what it's about, what is the maximum, minimum value, the range of values. And here is, you can see is how this raster has been displayed, okay? Now, just to give you a sense of how this raster stores data, you see this inquire cursor here. If you click on that, and you click on any place in the raster, like, okay, let's click here on your Mysore. Yeah, so first you have to select this before you click, because whatever layer is selected in this dialog, it will give you the information on that in this box here. So you can see that for that pixel, the mean annual temperature is 24 degrees. Okay, so this is how you can query the raster. If you right click here, you see there is no option to look at an attribute table because there is none. Yeah. Okay. So any questions here on rasters and data format for raster? Because it's a raster. So if you remember to that slide, think back to that slide that I showed you of how the raster stores data. There is one attribute value per pixel. And the way that in QGIS you can find out what is that attribute value is you can indirectly just go to that inquire cursor, it will give you the value here, see 24 degrees, yeah? And later on your own, you know, I encourage you to play around with the symbology, so you can change how rasters are shown. Instead of using grayscale, you can use a color ramp, things like that, yeah? But any questions on rasters? Do you all understand how the raster stores information? Yes. Yes. So thank you for pointing that out. So here you, as he says, now you can't see the place points, right? So anytime you bring in a raster, if it's at the top of the layer dialog, because it's a continuous surface, it hides any vector layers that are under it in the layer dialog panel here. So the way to visualize this information is to pull the raster to the bottom. You can just drag and pull it down here, yeah? And now you see, because your polygons are displayed with a fill, the symbology of the polygons, the country boundary has been displayed with a fill, you're not able to see the raster under it. So the way to change that is just go here, right click, right click on countries, go to properties, go to symbology. You can change this to from a fill to transparent fill. See this here? If you tick that, it will remove the fill, and you can change this line to something that will stand out, maybe in cyan. Yeah, and apply. Okay, yeah. So now the country boundary is in this bright blue, cyan, and it's showing you the raster values. So if you are going to make a map, you would have to play around with the symbology here and bring it to something which actually you can see all the information on the map. Any questions? Yeah. So I'll just go back to what we were doing before we went into QGIS, which is, I still need to tell you all about what to watch out for with when you're using GIS systems. So basically there are different types of error that you can have with spatial data, yeah, and any kind of data for that matter. But you can have noise around the actual value of the data set, that point in space. You can have positional error, okay? But you can also have a shift. So this very often happens with GPS data, is if there's an error, it's bouncing, the signal is bouncing off some kind of topography or something, it will shift everything by a certain, there'll be a systematic shift. That's called a bias, okay? So sometimes you will have this noise, this sort of positional error noise plus a bias, a systematic shift in one direction, okay? And so this green star here is your actual position. Yeah, that's the actual position. Here you have noise. Here you have noise plus bias. And here you have a situation where there is not only noise and bias, but it's actually telling you there's an inaccuracy. It's giving you a reading that the position is somewhere here. So that can be some error related to how the data was gathered. Okay? Yeah? So just to keep this in mind, is that all GIS data sets have some level of error in them. A good metadata will try and quantify what those sources of error are. Yeah? So this is just to show you how the difference between accuracy and precision in the context of a GIS. So say here I have a aerial photograph, and I'm trying to identify forest cover, different types of forest, not just forest cover, from this aerial photograph. So I have different, I'm sorry, his color is not showing up here, but I have different interpreters looking at this, interpreter A, B, and C, 3 people, trying to interpret the image to say, okay, this is this forest cover type, this is this forest cover type, and this is where it is located. So each person will have some error, okay, in their interpretation. So you can superimpose those boundaries to get sort of a boundary that averages across that, okay? Now precision in the context of a GIS is really to do with how precisely you define where in space that object of interest is, okay? So precision deals with delineation or where in the context of a GIS. Okay, so some things can be precise, very highly precisely defined in terms of spatial position, and some other things less so. When it comes to accuracy, so interpreter A says this vegetation patch is this kind of forest, cottonwood trees. Interpreter B will say, no, this is actually ponderosa pine, okay, stand. And interpreter C will also say, okay, it's cottonwood, okay. So when you deal with accuracy, or in this case, classification accuracy in a GIS, it's to do with the question of what rather than where, okay? Okay, any doubt? So when you talk about data being precise or less precise in a GIS, it's how well the spatial location is defined. When you talk about accuracy in a GIS, it's how accurately that identity has been derived, okay? So this is accuracy and precision in a GIS context, yes? Yeah, so this can be done with aerial photographs. Often you use visual cues because it's very high resolution, so you can actually see the texture of the canopy. Now, of course, there are also algorithmic ways to do it using object-based classifiers and so on. But typically, traditionally, with aerial photographs, a lot of vegetation mapping would actually be done with some ground truth data. So you have GPS locations in some areas of, okay, this is what a pine stand looks like. This is what a cottonwood stand looks like on the ground. You've collected this data using GPS on the ground, and then you superimpose it on the image, and then you see, okay, on the image, this is what pine looks like. On the image, this is what cottonwood looks like. But then you have areas where you don't have ground data, right? So you're using that contextual information that you've gathered from the ground and interpreting what's in the image. But in that process of interpretation or classification, there is error. And that is an error of accuracy. not an error of precision. Yeah? So there are two kinds of things in terms of what can generate error in any given spatial data set. The accuracy of the data set and the precision of the data. The problem with GIS, because it has got this fluid scale, you can zoom in, you can zoom out, it nicely shows you one layer on top of the other, it gives you a false sense of precision. So you can have, so all features are stored with precise coordinates, right? The computer stores some numeric value of the X and the Y, regardless of the precision of the original data source, okay? So say you have two maps. Oh, okay, think this, here you have a case where you have these two coordinates, here, and one is more precise than the other. This one is more precise than this one, right? But in the GIS, you'll see the point in the same place, right? So this sometimes gets lost. That one data set, when you bring in many data sets from many different sources, sometimes we don't think about, okay, how is that data generated? At what level of precision was that data generated? So this is something we need to always keep track of. And the easiest way to keep track of it is through a good metadata, if that is available. If that is not available, then you just have to, that's the problem. You can't actually understand the level of error in the layer. So depending on the scale at which you view these points, so you can zoom in and zoom out, right, in QGIS, they will either look like a single point, or if you zoom right all the way in, they may separate, right, based on the fact that the two are separate. So as you zoom closer and closer, the relative distance between the points will increase. But this is very hard to do in any given analysis when you're dealing with hundreds of points. Yeah, and you're dealing with different types of layers, like you saw, you have a country polygon and a point polygon, and then you have elevation or you have temperature. It's very difficult to check everything. Okay? So more importantly, you need to remember that if you're using data that was generated at many different spatial scales, so if one data set was generated at a global scale, another data set was generated at a taluk level, there will be differences in precision, okay, because of the scale of the data set. So this does introduce a mismatch in precision levels in the analysis that we need to keep track of. Yeah. So importantly, very importantly, know your data sources. Data from any mixture of scales can be displayed and analyzed in the same GIS project, but then there are these things to keep in mind. Since scale is fluid, features may appear more precise than the data that they're actually based on. So always be careful to note the original source scale for the features from which you got the data set in the GIS. So this is just to give you an example. If I'm creating a layer of, say I'm trying to digitize the boundary of this inlet water body, yeah? If I zoom out to 1:100,000, I showed you that scale bar in the QGIS. If I zoom out to this scale and I digitize that boundary, I'm going to get this kind of coarse thing, right? But if I zoom to this scale, I end up with a much more nuanced contour here, which has all the inlets and bays and so on, right? So sometimes this information is not available, but when it is, please use it so that you know how precise, this is dealing with precision, how precise the data set is. So also, very importantly, when you mix information from different scales and different sources, this will generate error in your analysis and therefore may affect your conclusions that you can draw. So always stick by this rule. The chain is only as strong as its weakest link. So the accuracy and precision of measurements, maps, and models from a GIS are only as good as the least accurate and precise data source, okay? Because you cannot generate new information that doesn't exist. So that weakest link is actually the level of the precision and accuracy of your entire analysis. Yeah? Any doubts on this? So I finished my slides on GIS, and I was going to do another session on how GIS can be used in epidemiology. So that's the next, sorry. Yeah. So now we'll talk about how we can apply all of these tools. So just so that we're on the same page, I know you had many lectures on this, but for the purposes of this talk, I'll be using this definition of disease ecology. It's the study of host pathogen interactions within the context of their environment and evolution, understanding pathogen transmission and spread over space and time. So you have both space and you have a temporal dimension and impacts on host populations. So this just shows you the different elements in a system where you have the pathogen, you have vectors, you have hosts, and all of them interact within an environmental context and over time. So this is a quote from Professor John Zellner, who's a spatial epidemiologist in the US, and he says that the reason space is particularly powerful for building a relational perspective in epidemiology and public health is that you can put anything on a map. Everything that is within the concern of public health can be pinned down to some location on a map. Whether or not that location is meaningful is another question, but at least it gives you a place to start. So going a bit further, this person Waller wrote in 2017, so disease ecology is very relational, right? It's all about relationships, one at the individual level, between hosts, across hosts and vectors, and so on. So the interactions of at-risk individuals with each other, with other species, and with elements of their shared environment, okay? Relationships. Accordingly, spatial factors often play key roles in disease ecology due to the influence of heterogeneous geographic distributions. So host distributions can vary, vector abundances vary, there are landscape barriers to contact on the spread of an infectious disease. So when we bring GIS into this, you can see that there is big scope here because it is based on these spatial relationships, especially things like spillover, when hosts and pathogens interact in the environment, leading to disease emergence. So one role of GIS is in minimizing the risk of future pandemics. Another role of GIS is it can be used for surveillance and monitoring of disease spread. It's very powerful in that. You can use it for modeling. So I think a lot of people have covered niche modeling. Yeah, so that's one way in which GIS can come in. Have you all has somebody talked about niche modeling with your ecological niche modeling? No? Okay, so ecological niche models is one example of the modeling. Then based on things like ecological niche models, you can use it to forecast how disease will emerge or how epidemics might behave, and where public health interventions can be implemented to contain a disease. Yeah, so designing interventions that mitigate, control, and potentially prevent epidemics and pandemics. So these are many different powerful ways in which GIS can be brought in disease ecology. I think this you have seen, this framework by Plowright et al. These are the pathways by which spillover can happen. And I'm not going to go into it because, should I go through this? Okay, fine. Okay, so I'm just going to tell you then, you can see that here there is something called pathogen pressure, right? Which depends on how host populations are distributed, how pathogens are released, how they survive, right? So here, GIS can definitely help in mapping this component of it, yeah? Also, some parts of this, survival and development. In terms of release, I think some certain things like harvest rate can be mapped spatially in a GIS and even over time. And certainly this aspect of human exposure how behavior leads to contact with the pathogen, things like wild bush meat, or even incidents of vector-human interfaces can all be mapped. The gap here is I'm not aware of any GIS applications at this level within the host barrier as yet, but if any of you have come across anything, that would be really interesting to hear about. Yeah? But as you can see, like many places where GIS can come in. So this is, again, I don't know if you've seen this, but this GIF just shows you how spillover happens. The take home is that these gaps in these barriers are very dynamic in space and time. So you need a tool like GIS to be able to track over space and time where that gap is happening. So when an event happens, if you have enough data in a spatial context, you can actually pin it back down to a spillover event that happened at a particular place at a particular time. Yeah. So a very, very fundamental part of disease ecology, GIS plays an important role. Yeah, so these ticks just show you at all of these different barriers can be looked at and mapped spatially and temporally in a GIS framework. Yeah. Okay, so going at a more fundamental level, this is a paper by Ansil et al. Published in PLOS. They used GIS to design their sampling. Okay, and what they had was a land cover map, which was a raster probably. It could have also been a vector, I'm not too sure. And so since their question was to look at small mammal associated bacterial communities in mixed use landscapes, and they wanted to make sure that they had enough sampling based on the distribution in the area of each land cover type. Okay, so here you will have a GIS. In a GIS, you'll have a layer with your land cover types. From that, you can calculate the area. And based on that, they could decide, okay, this is where we need to sample. And this is the intensity at which we need to sample for each land cover type. So this is an example of how GIS can help you design your study. Of course, here you can also see that it has come in at the communication stage where they've built this really nice map and put it as a figure in the publication. So at both ends, GIS is useful. Yeah, so one very important way in which GIS can help you, going back to the quote by John Zellner and Waller, is that by visualizing things, you immediately start to see patterns, right? That's the most powerful way to pick up on patterns. So GIS lets you customize spatial boundaries to whatever unit makes sense for your study. So you can customize it for specific study areas. So if you want to look at it, say, at the level of a taluk or at a level of an ecoregion, you can design your boundary to be at whatever scale or unit makes sense for your question. Okay. And once you have that boundary, you can use it to estimate underlying population counts and characteristics. Okay. So one tool is this is an example of what's called chloropleth maps. So chloropleth maps are basically aggregating over a particular boundary area, whatever that variable is of interest. Okay. And then visually you can look for patterns. You can look for gradients in anything like population counts, senior citizen distributions, how much poverty, where the poverty is, you know. So these types of maps, which are basically vectors, these are vectors, polygons, and each polygon is assigned a value in each of these things, and then you can, to show actually visualize it, you can bin the continuous variable into these categories that make more sense to you, and then you can visualize that to look at patterns over space. Yeah, any questions on chloropleth maps? No. Oh, you can also do some preliminary spatio-temporal analyses, which can provide evidence of unusual disease rates across time. Okay, So you can superimpose maps over time to see how things are changing and if you are seeing hotspots of disease emergence in certain areas over time, okay? Such visualization can yield information on potential transmission factors and how disease patterns may be changing in an area, yeah? Okay, so a little further into that from just some exploratory sort of visualizations over space and time, you can actually run spatial statistics in a GIS framework. One common use is, so you usually events of disease detection are depicted as points, right, in space. Okay, you have a point layer of where that case was. Okay, now based on these points, you can run various point pattern analysis techniques. Some of which I've mentioned is you can do cluster analysis, you can use kernel density estimators. And basically the questions you can look at here, you can test hypotheses on what's generating these patterns and risk factors, depending on whether patterns appear to be random or not. Okay, so in point pattern analysis, you're usually asking the question, is whatever process that's generating these points, is it a random process or is it somehow different from random? Okay, so that's a starting point with point pattern analysis. Now, kernel density estimators, anybody here used a kernel density estimator in their work? So what these do is that they take each individual data point and they build a probability distribution function centered on that point, okay? And then the kernel density estimate is the summation of all of those probability density functions across the extent of your data, okay? So you'll get a surface, a probability surface for your data set based on these point locations and how densely packed they are over space or how spaced out they are, right? So an example of this was, some of you may have read this paper by Warraby et al. In Science 2022, where basically what happened is that initially, people were saying that this Wuhan, this Huanan seafood market was the source of COVID, SARS-CoV-2. And then there was this whole big controversy where people started questioning that. So Warraby et al. Then went back, looked at the data from December 2019 that was published in a WHO report. And based on that report, they mapped about 155 cases, okay, in Wuhan. And this is a point layer showing you where those cases were. This square here is the market, the Huanan seafood market. And they distinguished between points that had a known link to the market and points that had no known link to the market, as in there was no way that those people had either visited the market or were in contact with somebody who had visited the market or anything, okay? Then they kind of did what I was talking about, a kernel density estimator. I'm just simplifying it very a lot, but essentially it was a kernel density approach. And they built a probability surface, right? Centered on relative to the market, okay? They looked at what is the probability of detecting both types of cases, linked to the market, not linked to the market. Remember, this is December 2019, okay? And what they found was that, okay, all the cases here, this is the surface probability kernel density estimate of all the cases, all 155 cases. You can see that the closer you are to the market, the higher the probability of a detection, okay? So this is the 1% contour from here. And you can see that here it's like, this is hard to read, but I think that says 300 meters, okay? This much is 300 meters. So we're well within a kilometer, okay? That 1% contour of probability is very close to the market. Then they looked at all the cases that had no link with the market, but were COVID positive. And they found that it's actually even more closely packed to the market. So the people that had no connection, there were many cases that had no connection to the market, that were closer to the market than actually the people who had connections to the market. So they use this to argue that this is evidence that this market was the point of origin of SARS-CoV-2, okay? And this they did using spatial point pattern analysis and kernel density estimates. Any questions, doubts? Okay. So, yeah, oops. The other piece of evidence that they used also from a GIS is that they mapped the market itself, okay? And if you remember that people had gone into the market and taken environmental samples and tested them for the presence of the virus. So they used that data to build this kind of heat map, yeah, of where were most of the positive cases located inside the market, right? You can see here there's another contour surface built on the point data. And so happens that this southwest corner of the market is where they were selling live animals, particularly raccoon dogs. They had evidence of this, photographic evidence of this. So they put all of this together to argue that yes, in fact, this market was the origin of the pandemic, and it was linked to this illegal wildlife trade that was happening, sale of wild mammals that was happening in this corner of the market. Yeah. So all GIS based. Okay, so that's just looking at spatial patterns with the disease data itself. Now there's a whole bunch of other information that you can bring in, which we call contextual information. So this is supplemental data that add another dimension to the analysis, enabling further contextualization of the public health problem. So for instance, here you have a very high resolution satellite image of a neighborhood in South America. And what people have done here is they're looking at some mosquito-borne disease, and they're trying to map all the stagnant water bodies in this neighborhood, okay? So bringing in contextual data. It can be done at this scale, which is very fine scale, where you can see individual houses and swimming pools and so on. Or you can do it, at this scale. This is all of Brazil. So in this study by Skinner et al., they looked at, I think, 3 or 4 different vector-borne diseases, malaria, leishmaniasis, Zika, dengue, chikungunya, all of those. And then they created something called a human footprint index, which was an aggregation. They mapped all of this, environmental infrastructure, transport networks, water systems. They used that to aggregate it to develop a human use index of the whole state, the country. Then they also used information on population, temperature, probably mean annual temperature, and percent forest cover. And when they put all this information together, what they found in a GIS is that certain types of vector-borne diseases like malaria and leishmaniasis tend to occur in areas where there is a lower human footprint. Whereas you move in this gradient from low to high human footprint, the type of vector-borne disease that you find changes. So Zika, chikungunya, dengue, very high here. Yeah, also with forest cover, you can see there's a gradient. So low forest cover, high human presence, the type of vector-borne disease that is most frequently encountered changes. So this has implications for public policy, yeah, and how to design public policy. So also the point I was trying to make here is you can work at many different scales, at the scale of a neighborhood, at the scale of an entire country. You can aggregate this contextual information. So you guys have heard of Lyme disease, right? Transmitted by this bacterium called Borrelia burgdorferi. Now there's this very interesting link between forest fragmentation, which is something we ecologists use GIS a lot to study. We use it to quantify how fragmentation happens over time, how the extent of fragmentation, the intensity of fragmentation, and the type of fragmentation can all be quantified in a GIS context. We can do things like quantify loss of forest cover, quantify spatial patterns in loss of forest cover. Is it all happening in one place or is it distributed throughout? We can use it to quantify sizes of remnant patches, how much edge there is around remnant patches, how irregularly shaped these remnant patches are. We do all of this in a GIS, okay? So as an ecologist, forest fragmentation is something that I have used GIS a lot to quantify. Now here, researchers have found that this process of forest fragmentation is integrally linked to the spread of Lyme disease, and this is the mechanism. And the way that they came to this understanding is, again, through the use of GIS and mapping. So they looked at the loss of forest, and they found that the loss of forest was associated with the removal of predators. Once that predatory pressure was removed, they found that there was a five-fold increase in one of the most competent reservoirs of this bacterium, which is the white-footed mouse. Okay, and so they found actually that the abundances of white-footed mice were much higher in fragments that were less than 5 acres in size. Okay, so and that was typically the kind of remnant patch that you would find in the suburban US kind of landscape. So this is actually how fragmentation is linked to the spread of Lyme disease, which is now one of the dominant vector-borne diseases in North America. Okay, so that was understanding ecological processes and their links. Now we can also look at how to map connectivity. So disease spread and persistence is an important area of research. In a GIS, you can look at how spatial structure and landscape level heterogeneity can influence the spread of pathogens, which in turn, that depends strongly on what's called a dispersal kernel. Have you all heard that term, a dispersal kernel? It's basically, if you think of a histogram, a distribution of the range of distances that a pathogen or a vector can travel. Okay, so that histogram is called the dispersal kernel. So you can use that information in a GIS with this information on landscape level heterogeneity and try and look at what implications it has for disease spread. Yeah. So what people have found in some cases using this approach is that often localized movements can give rise to what's called traveling waves of infection. So you have peaks of infection over space that are time followed by troughs. Examples of these, it's been shown for rabies with raccoons, measles and influenza in humans, and baculoviruses in insects. So it's just a graphical representation of, so you have time on this axis, and you have spatial position, and then you have these traveling waves. Okay, so this is from the study on measles by Grenfell et al. From England. And again, using very strongly based in spatial analysis, using a GIS framework, they used something called wavelet analysis to come to these inferences. Another framework that is often used to look at connectivity and persistence of pathogens in a landscape is host parasite metapopulation dynamics. And if any of you have worked in ecology, you know that metapopulations are inherently spatial in structure. So you have these patches, and then you have connections between these patches, and hosts and pathogens move among connected patches, okay? But what leads to persistence that people have found is that even a small amount of host movement across patches increases the long-term persistence of both hosts and parasites in a landscape by allowing some individuals to escape heavily parasitized areas. Yeah? Another sort of special case of connectivity is something called network analysis. So you may have heard of social network analysis. This is more a spatial network analysis, which you can do in a GIS. So an example of this is you can use public transportation networks to analyze things like travel times, distances, functional distances, and general connectivity, especially with human populations if you're using a road network. And this is an example of a set of maps that was derived after Hurricane Maria hit Puerto Rico. They used network analysis to demonstrate, to look at the loss of connectivity to pharmacies after this event. So here's the initial state. The areas in gray hatching show those which are within a 5-mile drive of an open pharmacy. Then you had the hurricane making landfall, and 10 days after landfall, this is the situation where you have this loss of connectivity with pharmacies in a large part of the central part of the country. And that's just the inverse of that is mapped here. So you can actually pinpoint these areas which lost access and then design public health interventions based on that, or emergency relief based on that. Yeah. So so I mentioned ecological niche models before. Should I go into, yes, no? Briefly, okay. So the way niche models work is that you will have a set of occurrences, points. This can be the presence of an organism or the occurrence of a disease or something. Right? It's a series of events. You have points. You can use in a GIS, you can use these points to derive contextual information about those point locations.
Arundhati, we have not actually discussed this thing. We only discussed occupancy modeling.
Oh.
Yeah, but not really niche modeling. So it's okay. It'll be interesting to the group to be.
Yeah, so yeah, niche modeling, I see a lot of application of niche modeling in disease ecology. So essentially, this is how ecological niche models work: is you have a set of points, and then you use that point layer in a GIS framework to query associated environmental variables that of interest. They have to be somehow related ecologically or to those points. So the examples of this can be things like climate, temperature, rainfall, topography, human density, land use, okay? All of that information, usually it's in the form of rasters here, okay? So you have a vector layer, you have raster layers here. You extract information on the raster layers based on your vector layer in a GIS, then you have a data set, and you use that data set, and there are many ways of building niche models. You can actually use like statistical approaches like logistic models and so on to build a niche model. Or you can use so have you heard of Maxent? Yeah, so you have these other modeling tools which basically use certain probability rules to and give you a map. And that map, that output is a probability of that thing of interest across space. And if you're looking across time, you can get it across time as well, okay? So in a nutshell, that's what niche models do and how GIS comes in with niche models, yeah? Any questions? No? Okay. So here's an example where niche modeling has been used to predict the geographic distribution of anthrax, okay? And this is the legend. Again, so the output from a niche model is usually a probability map. So you will have a color gradient to depict the probability going from 0 to 100. So the red here is high probability, yeah? So you can get outputs like this from niche modeling. It's very much within a GIS framework. Yeah, so niche models allow you to look at distributions of, in a disease ecology context, distributions of vectors, hosts, and pathogens, understand how that varies across space. And also with climate change, now there are layers available through WorldClim and so on, which predict future climate scenarios. So you can bring those in to project how a disease or pathogen might shift over time under future scenarios. So it just doesn't have to be climate. It can be land use change. It can be anything, right? So it's a very powerful tool. So one, to build models. Second, to use those models to forecast. A good example of this is this study by Carlson et al., published in Nature in 22. They did a very complicated analysis where they were actually trying to look at how in 2070, given the IPCC future climate scenarios, they used that to try and see how, and across 3,000 mammal species, they looked at mammalian hosts and the pathogens, and they brought in the phylogenetic relationships between those hosts and the pathogens, and using that information, and also information on land cover and climate, they built a model to see how often or how common crossover, what's the word? Viral sharing events might happen in the future under warmer climates, okay? So one thing they found is that especially in tropical areas, things were moving in response to climate change, would move upslope, particularly in these biodiversity hotspots. So then you were having these novel assemblages of mammals, and with the novel assemblages of mammal species, also a mix of pathogens, and so you can have these like new viral sharing events coming out. So what they did is they first mapped based on climate, how it would look in 2070, the probability of viral sharing events. And then this map, they brought in where human population centers would be in 2070 based on climate change and land use change. Okay, so like I said, it's a very complicated study, but you should take a look at it because it really brings forth the power of GIS. So here in this shading, you can see that this area is high probable viral sharing events and high population density, and you can see that India really emerges as a hotspot for that in the future, okay? Okay, so this is an example of an open source platform that is available. This is the URL, Health Heat Map of India. Okay, so a tool like this is incredibly useful for public health policy and interventions, yeah, because you can see almost real-time maps, and these are like those chloropleth maps, they're calling them heat maps, of where there are outbreaks of different things happening. Yeah, different. So here what I've shown is this respiratory infection ARI in the last 2 weeks preceding the survey. That's what's mapped here. Yeah. So this is a very interesting GIS, web GIS based tool. So you can take a look at it. This is another application in policy, public health policy for GIS. Okay, so just to shift gears a little bit. So I've talked, any questions on applications of how we can bring in GIS? Any questions on that? Okay. So the next set of slides, and I'm almost done, so I'll probably be done early, is just to tell you where to look for GIS data for your work. Yeah, so I haven't had a chance to get into remote sensing much. I didn't want to spend time on that because that's another whole actually lecture. But nowadays, remote sensing is a major source of information on GIS platforms. And you have two major types of remote sensing data. You have passive remote sensors, which essentially capture reflected radiance from the Earth's surface. That's reflected sunlight energy. And then you have active remote sensors, which send out a signal and then measure the reflection of that signal. An example of that is, you've heard of LiDAR, right? Another example is radar, microwave sensors, okay? So now these passive and active sensors, you all are familiar with passive sensors. So these are like just optical, like cameras which are fitted on these platforms that can just, like a camera does, record a signal of reflected light from a surface, right? So optical sensors work in different wavelengths. Most of them capture the visible light plus infrared, yeah? And you have hyperspectral sensors, which differ from optical sensors in the sense that their radiometric resolution is very high. So with most optical sensors, you'll have wide bands. You know, you'll have one band for green, one band for red light, one band for infrared, or 3 bands for infrared. With a hyperspectral sensor, like the bandwidth in which it's recording information becomes very narrow. So you have many, many, many spectral channels within which information is being recorded. So that's why it's called hyperspectral. Okay, and a lot of the application, at least in ecology for hyperspectral data, is some studies have actually used hyperspectral information to distinguish between individual species of trees in a forest patch. Like it's that fine scale, the information that they're recording is that different species based on their physiological characteristics reflect light differently. Their leaves reflect light differently. So hyperspectral sensors can actually capture that information, whereas a normal optical sensor doesn't have that resolution and bandwidth. And then microwave is also passive and active. So this is just to show you the range of platforms in which these sensors can be mounted. And as you go up the gradient, so of course you have the ground-based sensors, which will only look like at a few 100 meters around. Then you have drones, you have small UASs, you have unmanned airships, helicopters, airplanes, something that goes into the stratosphere. Then you have the satellites, which orbit very high above the Earth's atmosphere. And so the higher the platform is deployed, the larger the ground coverage. Yeah, that's the relationship there. This is just an interesting graphic that shows you all the different ways in which remotely sensed data can be gathered and then brought into a GIS to then make these different thematic sets of information like topography, wetlands, population density, land cover, and so on. Okay, so that's the relationship between how remote sensing comes in with a GIS. Okay, so where do we get this data from? When it comes to human data, there are a number of sources. There's one open access spatial demographic datasets platform. This is the data set that was used in the science paper that I showed you, www.worldpop.org. We can quickly see if that can go into that. Yeah, this is the site. Yeah. So you can see that the different parameters that they have information on, yeah? These are open source data. This is on world population. Then you have another well-known data set, which is housed in Columbia in the US. It's put together by NASA CDAC. This is another good source of demographic and socioeconomic data. Yeah, so this is a CDAC website. Yeah. So there's a whole bunch of data sets. Particulate matter concentrations, nitrogen oxide, urban rural population, land area. Yeah, so you can go through these sites when you have time. And this is a very important source of information on any human, social or cultural data source. Have you heard of OpenStreetMaps? Yeah. So here you can get information on pretty much anything, built environment, forest, lots of different things. Yeah. So, and there's a way to actually bring this information in from the web server into a QGIS. Same way you can also bring in Google Earth satellite data as a backdrop in QGIS. Yeah. So yeah, we can see your roads, forest areas, rivers. All of this data can be downloaded, yeah? So if you zoom in, you can see like even individual houses, structures, roads. It's very fine scale data, yeah? Reservoirs, lakes, all of it. So OpenStreetMaps is a really nice source of information for GIS data. Okay, so on species, you've heard of the Global Biodiversity Information Facility, GBIF, yeah? So GBIF is usually, Ansil, please correct me if I'm wrong, but is that the go-to place for species occurrence data now? Still
GBIF, but what in practice, a lot of people use a IUCN list for the distribution data.
Yeah.
And also people started using, yeah, you already mentioned, yeah, okay.
No, please finish.
Now, places like iNaturalist and
Yeah.
things like that, yeah. Okay. Sorry.
Yeah, so GBIF is just point data, right? You can get lat longs from GBIF. As Ansil mentioned, from IUCN, you can get range maps, range map information. You can also get other information like what are the threats, what are, so IUCN focuses on vulnerable and endangered species. It's not got everything. Another thing that I found very useful is the Global Invasive Species Database, GISD, which will give you the geographic locations of where some of these invasive species are distributed across the world. And those can be brought into a GIS or translated into a GIS based on the information that's there. CABI, CABI, what does CABI stand for? Do you remember? CABI, CABI. I've forgotten what it stands for, but that is another source of information on invasive species, yeah, if that's of interest to you. India Biodiversity Portal has a lot of data on the Indian context, both species data, forest data, land cover data, hydrology data, all sorts of interesting things. They had a WebGIS interface as well. I'm not sure if they still do, but that is worth looking into. And then, of course, iNaturalist. How many of you have been to iNaturalist, have used it? Not many, but that's a very good source of information. It's basically a citizen science operation. And so you have many records for a wide range of species across many taxa. Yeah? Anything else, Ansil, that I've not covered with species? Okay, so now you can also get a lot of satellite imagery, especially from NASA. And also now the European Space Agency Sentinel is a satellite. Are you guys familiar with the Sentinel satellite data? It's actually quite relative. So the Landsat data, that entire Landsat archive is available from NASA. And Landsat was first launched in 1970s. So there's a really nice time series going all the way back to the early or mid 70s available through NASA, through their Landsat series. Now that data is about a 30-meter pixel resolution. But more recently, you have the Sentinel series from the European Space Agency. That pixel can go between 20 meters to even 10-meter resolution. So you have much higher resolution data available in the last decade, more or less, for all over the globe through Sentinel, okay? So if you go to sites like the USGS Earth Explorer. I'll just take you there through this. Yeah, so this is the interface. You have to sign up, you can register, and then you can freely download data. There's all sorts of satellite data available from all the NASA satellites. You may also get Sentinel data. Then there's other data available, like things like fire occurrences, vegetation, land cover, NDVI, yeah. Okay, sorry. So remember when I showed you the picture of the raster and I said, okay, what determines the size of the pixel? It's the rows and the columns. So there are two things with the raster. There's the extent of the raster, so how much of the Earth's surface it covers, and then there's the resolution. How big is the pixel? So each pixel in a Landsat image covers an area on the ground that's about 30 meters by 30 meters, okay? So in that 30 meter by 30 meter area, it's picking up the reflectance of many different things, right? Within that, in a 30 meter by 30 meter area, you can have a house and a lake and a tree, and all of that is giving a mixed signal back to the sensor at a 30 meter resolution. But if you reduce it to a 10 meter by 10 meter resolution, then you're getting a much clearer signal back from individual objects on the ground or features on the ground. So high resolution data is always nicer, but it depends on the question because there's a trade-off, right? The higher the resolution, usually the smaller the extent of the image, and the heavier the file, the data file. So it becomes difficult to port these very high resolution images. Usually people don't do very high resolution analysis at very large spatial scales. There's a trade-off between spatial scale and extent. Yeah. This is a good place to go and explore what satellite data is available through NASA and the American system. Okay, for climate, I've already mentioned WorldClim. WorldClim is a set of climate parameters that basically represent the average between, I think, how far back does WorldClim go? I don't remember. I think it's around, it's the mean between 1970 and, no, it goes further back than that. Anyway, it's an average mean over a time period of many decades in different things like temperature, rainfall, and it's available for the entire Earth surface. So I showed you that raster on mean annual temperature that you saw and opened in QGIS, that I downloaded from BioClim, okay? So BioClim is nothing but these WorldClim parameters synthesized into a way that's biologically meaningful. So things like annual temperature range, minimum temperature of the coldest month, things that might affect physiology of living beings. So that's what BioClim is. So it's a set of 15 parameters which you can get for the whole Earth at a 1 kilometer by 1 kilometer scale. Okay. More recently, there's another data set called CHELSA, CHELSA, which I haven't listed here, that also gives these bioclimatic parameters. Yeah. Okay, another immense resource, and though some of you have worked with this, is Google Earth Engine. Here, so if you've not used Google Earth Engine, I encourage you all to sign up, get an account, explore it, because it's extremely powerful. And I was talking about the democratization of GIS. Now you have these immense data sets at your fingertips. All you need is a good internet connection, and you need a knowledge of Java and scripting to work with it. But I'll just show you the range of information that's available on Earth Engine. So I'll just scroll through, you can take a look. So DEMs are digital elevation models. They're a representation of topography. So you can get hold of DEM data, you can get hold of satellite data. There's this very interesting data set called nighttime lights available for the whole planet. And this actually is a good proxy for many human-related issues and impacts of humans on natural environments. Night lights is a really good proxy for that. See, there's like other products which have been derived from Landsat imageries that are also available on Google Earth Engine. You have land cover maps, you have demographic data, yeah, basic demographic characteristics, gridded population of the world. These are all freely available. So you can just go through the data sets available. Then these are the different satellite archives that they have. They have the entire Landsat collection. Okay, all the Landsat satellites, all their Earth observation satellites are available through Google Earth Engine. MODIS is a coarser resolution platform sensor, but its advantage is that the temporal frequency of how often it revisits the same place on the Earth's surface is very high. So if you're doing any kind of temporal change analysis, MODIS becomes very useful. MODIS has been used to track green changes in NDVI. Have you heard of NDVI? It's normalized difference vegetation index. So based on satellite data, you can compute different indices. Like in the case of vegetation, NDVI is commonly used. And NDVI helps you understand how much green biomass there is in a place. So that's what NDVI tells you. So it's very good for looking at vegetation and changes in vegetation. Then MODIS can also be used a lot, has been used a lot for mapping fire occurrence, characterizing fire regimes. There's an archive of Sentinel data, which I was talking to you about from the European Space Agency, yeah? So do look at Google Earth Engine. Another set of data that's very useful, I think, for disease ecology that's available again through Google Earth Engine, but I've listed it separately because I want to draw your attention to it, is this is a data set that was first published by Hansen et al. It's run out of a center in the University of Maryland. They are tracking global forest cover across the world at a fairly high resolution, fine scale. And you can also get very fine scale maps of forest cover change. So where forest has decreased, where it's increased, and you can think about all like the useful things that you can do looking at especially wildlife, pathogen, human exposure, and because a lot of spillover happens when people deforest and move into like recently cleared areas, right? So I just separately wanted to draw your attention to these data sets on global forest cover. Apart from that, more generally, there are a lot of land use and land cover data sets available. Now, these are basically vectors based on satellite image interpretation. So they've used satellite data to analyze it and then map land cover, but the output is usually in a vector format, okay? But not across the board, but mostly you will get vector maps of land cover. So ISRO has a website, a platform called Bhuvan, where a lot of GIS data is available. And they have land cover maps. They do one every 5 years. So you can actually track change in land cover over time based on their products, yeah? So this is very useful. I mentioned Survey of India topographic maps. I want to show you that because topographic maps for many of us is really the starting point for the go-to place for information, especially in India. So Survey of India has a series of maps called Open Series Maps, and these can be downloaded, okay? They also have, political map of India, boundary, physical map of India, road map, railway map, all of these things. Oh, they do have a world map. Also by state, they have maps. Now in topographic maps, you get a lot of different information. You get information about all kinds of human features. You get information about administrative boundaries, both forest administrative boundaries and district administrative boundaries. You get information about anything from temple locations to tanks to houses, individual houses, village names, all of that is available on a topo sheet, yeah? So usually we go first to see what topo sheets are there when we do GIS. Yeah. And you get, I should mention that the topo sheets you get are different spatial scales. So you get very coarse scale topo sheets that cover a larger area. So that's 1:250,000 scale. Then you have 1:50,000 scale, which covers smaller like parts of a district. And then at a very fine scale, you'll have 1:25,000 maps. So there's a whole series of different scale maps available with topo sheets. This is the UN FAO global land cover map is also there. There's a map derived from MODIS. I mentioned the MODIS satellites. Then something called the Climate Change Initiative also has a land cover map available globally. And finally, this Esri is the makers of ArcGIS. They have land cover maps publicly available. Yeah. So there are many sources of land cover. The reason is that everybody defines land cover differently, and the things that you want to map in land cover may differ from one person's classification to another. So often you might have to go to 2-3 different sources to find the map that suits you the best. Yeah, I'm done. So, any questions? Yeah. Maxed out. Yeah, you have a question? I didn't catch you. Street map?
So in QGIS, can we view a map for streets in a street view, like as if like open street map?
I'm not sure. But there is a mechanism to link it to that, because there is a way in QGIS. So the way you bring in a Google Earth image in the backdrop is through the XYZ tiles. And maybe if you use the URL for the street map view, instead of the Google Earth URL, it will bring in that. So you can try that. I've never tried it. You can try add XYZ tiles and try linking. She's talking about street view, not OSM. OSM you can bring in. OSM street view you can? Okay. Yeah.
Similar to data sources, we have now different softwares as well.
Yeah.
So would you say QGIS is a great place to start, or like how different is ArcGIS? I mean, apart from that, it's paid. Would you recommend?
ArcGIS is much more powerful than QGIS, especially for rasters. But because it is so expensive, I wouldn't recommend it unless you have a very specialized need for doing some very fancy stuff. A lot of stuff can be done in QGIS, especially basic GIS analysis can be done, especially with vectors. QGIS is very good with vectors. It has some raster capabilities, but I think R, now a lot of GIS is being done in R. So if you have the right R packages, I can pass those on to you if you're interested. You can do a lot more sophisticated raster-based analysis, especially things like spatial statistics and all, through R. Much more than what you can do in QGIS. And what I usually do in my work is I toggle between things based on their different capabilities. So there's stuff that I like to do in Google Earth Engine, specifically if I'm working at very large spatial scales. Then you can import outputs from Google Earth Engine into QGIS or into R. So what I would say is there are now everything you get an open source version of, okay? So use the strengths of each, leverage the strengths of each in your analysis to do what that does best. So Google Earth Engine, really good for, you have access to all the satellite data, all these large scale data sets. You want to do large scale space-time analyses, Google Earth Engine is the way to go, yeah? It's just not very user-friendly. You want to do some mapping, you want to make maps, you want to do some vector-based GIS, QGIS is very competent. Yeah, QGIS is nice. You'll be able to do whatever you want to do in QGIS. You want to do some more statistical analysis, including ecological niche modeling and spatial statistics, things like that. R is a very good place. So And if you're not working at very large spatial scales, because it's very difficult to work in large spatial scales in R. So if you're looking at like a smaller landscape, like about 100 square kilometers or a few hundred square kilometers, or even maybe a couple of states, R can handle it, like sophisticated analysis at that scale on rasters. Yeah. So what I would say is find the strengths and go to those platforms which do it the best. Yeah. Between these three, you should be able to get everything you need. Yeah.
Hello. So my question is in regards to SDMs, and so we can build models as per past predictions, past variables and present variables. But when it comes to future variables, like where do we go? Because there are very few variables for future, I believe, and I'm not really sure. But how do we then compare? Because all our models are based on correlation. And if past and present would be fine, but future, if you build it through a different correlated variable,
Or you could
then
have novel environment.
yeah, so then how do we even compare? And where do we find future variables? That also.
So that is easier to answer. I think WorldClim has future climate data sets. So that's one possibility. I think the IPCC, usually a lot of what these climate change studies end up using are the actual IPCC scenarios for different, based on different, they call them RCPs, something climate pathways. So those I think are available. So look into the IPCC data sets for future climate. You can also look at some of these studies like the Carlson study that I put up. They have used future climate data, so you can look at where they got it from. I'm pretty sure it's IPCC data sets. This question about, yeah, I don't know. I think, see, personally, I used to use niche modeling a lot, but there are a lot of issues with niche modeling, and because of those, I've kind of moved away from that approach. But I see that it's still being done a lot, and it has its place. But I would say that niche modeling is useful as a sort of, in some way, an exploratory tool to kind of build a hypothesis, and then you have to take it further from there. You can't just build a niche model and say, okay, this is what's happening, like I'm done. No, because there are a lot of issues like you brought up with what kind of data it's built on and how those relationships are articulated. Things like Maxent is a black box, actually. Not a lot of people understand what Maxent does. So I've kind of moved away from using niche models as an end in itself. So yeah, I think for your question about future, Ansil, do you? Yeah, sorry, Farah.
We have used for parasite development rate for several parasites, this Plasmodium and all in Himalayan region. So what we did was we recorded the temperature for full year using the data loggers. So we had this is at different elevations, so you know like what resolution data you have, and we did very fine scale. I think we showed it also in we didn't go in the detail of that, but also from the world climb, we got the data from the past, but also for the future, which was very good to model in order to understand how good it is going forward and what would be the range of expansion of the parasite given the temperature fluctuation. The only drawback was that we needed the good half an hour resolution data, which world climb didn't have, because parasite development is very sensitive to change for 1 day or 2 days. If there is a block of 10 days, so it can be a arrested kind of abort development. But it works very nicely, just broadly, as you're saying. But in terms of really finding the niche and all, I don't think it will. But for a lot of parasite, I think models say for dengue and something, it should work very nice. And I think it's good to validate if you have your own data to understand.
Thanks, Farah.
Thank you. One more thing. So what did you move on to if you're not doing niche modeling? Just
Moved on to more of a less modeling, actually trying to collect data that, on the ground or from remote sensing to actually test hypotheses rather than build a model and sort of project that as the end of the story, right? So I'd say if you're using niche modeling, use that as a starting point. Use that to frame the hypothesis. And then, like Farah was saying, there are many ways in which you can collect information that can then be used to validate the output of the niche model or see where that niche model is actually working and where it's not working. Yeah, but I've kind of, in my work, I still use a lot of GIS and remote sensing, but I guess the scale at which I work has shrunk because now I work more at a landscape scale rather than a regional scale. Yeah, so I try to base my work in sort of bring in GIS and remote sensing as a tool, but not the main big powerful tool. It's always underpinned by field data. Yeah.
got it. Thank you.
Yeah.